At rest, the cell membrane of an axon is electrically charged so that the inside is negatively charged with respect to the outside. The electrical charge difference between the inside and outside of the cell is approximately minus 70 millivolts and is called the resting membrane potential. The resting membrane potential is maintained by a specific distribution of ions inside and outside of the cell. There are more sodium ions outside the membrane than inside. The opposite is true for potassium ions. There are more inside the cell than outside. A depolarizing stimulus changes the ion permeability of the cell membrane. The stimulus causes voltage-sensitive sodium channels to open, which allows sodium ions to rush into the cell. The inside of the cell becomes more positive, and the cell membrane depolarizes. After a slight time delay, the depolarization of the cell membrane causes the opening of voltage-sensitive potassium channels. The positively charged potassium ions diffuse out of the cell. Because the potassium ions are positively charged and diffuse out of the cell, the inside of the cell membrane becomes less positive. This is the process of repolarization. The voltage-sensitive sodium channels close during repolarization. This rapid sequence of events involving the depolarization and repolarization of the cell membrane is called an action potential. Sodium ions are pumped out of the cell and potassium ions are brought in by active transport. The resting membrane potential is reestablished very quickly. A myelinated axon is characterized by the presence of a discontinuous myelin sheath made by Schwann cells. Regions where the cell membrane is exposed to the surrounding fluid are called nodes. A nerve impulse travels rapidly through the myelinated portion of the axon and causes voltage-sensitive channels to open at the node. Positively charged sodium ions diffuse into the axon and an action potential is generated. The nerve impulse travels rapidly through the axon and generates an action potential at the next node. Transmitting a nerve impulse electrically through the myelinated regions greatly increases the speed of the impulse, since an action potential, which relies on the flow of ions, must only be generated at the nodes. In review, a stimulus traveling along an axon triggers the opening of voltage-sensitive sodium channels. Positively charged sodium ions rush into the axon. The inside of the cell becomes more positively charged and the cell membrane is depolarized. Voltage-sensitive potassium channels open and allow potassium ions to leave the cell, thus repolarizing the membrane. This depolarization and subsequent repolarization of the cell membrane is called the action potential. Once begun, an action potential propagates itself and travels along the nerve axon. Any change in the relative distribution of sodium and potassium ions that may occur during this process is counteracted by active transport, which moves sodium ions out of the cell and potassium ions in. Although impulses travel rapidly in this manner, their speed is greatly increased by the presence of a myelin sheath. Where present, this sheath insulates the axon and the impulse is transmitted electrically through the cytoplasm. An action potential is generated only at the nodes, and so the impulse seems to jump from node to node along the myelinated axon.